Pleasure to be here. So my talk today deals with two questions. What is life? And what is the smallest number of genes that one needs to build a living cell? We don't have the full answer yet, but I'll present what we have. So we couldn't really deal with those questions until the first complete genome sequences were done in 1995. The first one was Haemophilus influenza. It's the first time that we ever had a complete look at all the genes in an organism. There were 1,710 genes, 1.83 megabase genome. I recall in January of 1995, after we had finished the sequencing phase of this work, that Craig Venter and a few of us went to lunch. And Craig says, we need to do another genome fast so that people realize how easy the whole genome sequencing method is. So we decided on Mycoplasma genitalium, which is, was known as the smallest freely independently growing organism in nature, with about 485 genes. So we called up Clyde Hutchison, who had been working on it for several years, and I said to him, how would you like to have the sequence in 6 weeks? So a couple of days later, he sent 10 micrograms of DNA, and we prepared the libraries, and in fact, since it was so small, it closed off theoretically without any gaps, and we had the sequence in 6 weeks. So that was the first two bacterial genomes. And so this was the publication. They both came out about the same time. Well then, after those came out, Mushijian and Kunin at the NCBI had access to the sequences right away, and they came up with the notion that since they're both living organisms, the essential genes for life must be shared by each of them. So if they simply compare the two genomes, they would have this kernel of genes necessary for life. And they carried out that analysis. They had 234 orthologs, but in looking at the pathways involved, it was obvious that there were holes. So they came up with the notion of non-orthologous displacements. In other words, a gene that was not homologous or orthologous, but it had the same essential function. And this is something that now has complicated all comparative studies. So really most of these studies are, do not get at the question of the core of essential genes. So in 1999, Clyde decided he would put to the experimental test whether or not Kunin was correct with 256 genes. So he, for the first time, did a global transposon mutagenesis and arrived at about 380 genes that could not be knocked out. So there were 380 essential genes. And in the last paragraph of the paper shown here, he pointed out a couple of things. One is that a large number of the essential genes had no known function at that time. And secondly, he and Craig came up with the idea that we'd simply have to synthesize a minimal genome in order to ever answer the question. So, and this, I think, was spurred by the quote from Richard Feynman, if you can't create it, you're not going to understand it. So we figured if we could actually create a minimal cell, we might achieve some understanding. Well, what we needed at that point was the ability to chemically synthesize a whole bacterial genome, much larger than anything at that point that had been synthesized. And we needed a method to take the naked synthetic DNA molecule, the complete genome, and bring it to life, to install it in a compatible cytoplasm and boot it up, as a process which we called transplantation. And we also needed to identify genes that were essential for life. We had to have a very good method for that. So the first thing we did was to start synthesizing genitalium. It took about 2 years. We did it in stages and ended up cloning the complete genome in yeast. And it was sitting there. We isolated it out of yeast, but we couldn't bring it to life. It was just a dead DNA molecule. So concurrently, we were working on genome transplantation. We'd spent a lot of time with genitalium trying to get it to transplant. And eventually, we moved to Mycoplasma mycoides, which is a larger organism. It has over a megabase genome. We were successful with that, transplanting it into a closely related species, Mycoplasma capricolum. And the overall scheme is shown here. You take the purified intact molecule, and that's a fairly good trick to get a megabase without breaking it. And it was labeled with the tetracycline resistance gene. We then transformed that using fairly standard methods, polyethylene glycol and calcium ion, transformed it into recipient capricolum cells. And what we believe happens is that you get both genomes in some of the cells, and then when the division takes place, the two different chromosomes segregate to daughter cells. The ones that have the original capricolum don't grow in tetracycline, and they are killed. Whereas the one that has the donor genome begins to divide, and eventually you get a colony on the plate. And all the cells at that point have only proteins and other components that are derived from the donor genome. So we call that a synthetic cell. The three principal people involved are shown down here. And that work was published in August of 2007. Carol was the one that slaved away in a hood for 2 years on that. All right, so we could transplant mycoides, but we hadn't yet synthesized it. So the next thing we did was to put this whole genome together. And once we had it together, by synthesizing in several stages, starting with oligos going to 1 kb pieces, 10 kb pieces, and then the whole genome. The whole genome is assembled in yeast. We then isolated the genome from yeast and transplanted it successfully into capricolum cells. So we published this in Science in 2010. All right, so we had two of the things we needed. We could synthesize genomes and we could transplant them. The next thing we need, and so we chose mycoides as our target for minimization. It has a reasonable sized genome, a little over a megabase. It has 901 genes. The doubling time is just about an hour. and it gives nice colonies. So we were set up now to do a design, build, test cycle. We needed information now to design a minimal genome, which we could do in the computer, and then build the design, transplant, and then test to see if it worked or not. And you could repeat this cycle over and over. If this was successful, then you could make additional changes and go around. So at that point, we were eager to get going. This was around 2011. We didn't have enough information, but Craig said, let's just sit down at the computer, use the best knowledge you've got, and design a genome, and then we'll build it and see if it works. And he ended up about $1 million to do this. So we sat down, we used all of our knowledge of biochemistry and molecular biology. We had a little bit of data from transposon mutagenesis and from deletion analysis. So we came up with a design. We designed it in 8 different pieces so that we could test each piece to see if we had designed that piece correctly. And the red inner circle shows the pieces that we kept for the design. And this shows the relative size of the hypothetical genome compared to the starting CEN 1.0 mycotes genome. The thing didn't work. And, but one piece did work. Segment number 2, when combined with 7 wild-type pieces, did grow rather slowly, but it was sort of a foot in the door. And we learned a lot from that work. First off, we learned how to remove genes without disturbing. We set up a bunch of rules to remove genes without disturbing the expression of surrounding genes. We also got a lot better at synthesis. We semi-automated the process. We introduced error correction procedures, and we were able to build the genome in a matter of a couple of months rather than 2 or 3 years. We also, it focused us on the problem that we had to be able to identify the non-essential genes and the essential genes if we're going to make a proper design. So we decided to go with TN5 mutagenesis. This is a many transposon. Anything you put between the 19 base pair blue ends here makes up the transposon. And so we put in a puromycin resistance cassette, and we put in a couple of terminators on either end. And then we took the transposon DNA, reacted it with the TN5 transposase to make an activated transposon, or so-called transposome. And you then just introduce that into the CEN 1 mycoplasma cells by standard transformation methods and plate on a puromycin plate. And we got about 80,000 independent insertions. We scraped the plates and took an aliquot to pass serially for about 4 days. And at the end of the 4 days, all of the slow-growing mutations dropped out. So at the end, we had only vigorously growing cells, and any insertions in genes were considered completely non-essential. They didn't affect the growth. So we made DNA libraries of the P0 and the P4, did some inverse PCR, and then sequenced to find out where the insertions were. And we were able to classify all the genes fairly cleanly into 3 groups. Non-essential is a gene that doesn't get hit at all. Essential. Essential is one that doesn't get hit at all. Non-essential gets hit by both libraries, typically very heavily. And then the quasi-essential group are the ones that have a defect in growth caused by the insertion. And they show up in the first library, but are lost when you grow for several generations. And we found that 432 were non-essential, 240 were essential, and then this group here are the quasi-essentials. Since the growth rate is a continuum, we could break that into 3 different categories. So that potentially the non-essential and this IN group here are ones that we might consider removing, about half the total genes. So we were ready for the first design, and we came up with, here are the full lengths, 8 pieces. Here are the reduced segments and the fractional content. So it was a genome that was about 50% of the starting size. And we went ahead and synthesized that. The thing worked. All of the pieces worked. But when we combined them together, only certain combinations worked. And we picked one which seemed to behave pretty well, which we called RGD, that's Reduced Genome Design, segment 2, 6, 7, and 8. So it had 4 of the reduced segments and 4 of the wild types and 1 segments. All right. So we then took that and did another TN5 mutagenesis, and we found 26 genes that had gone from either essential or quasi-essential to, 26 original non-essential genes that had now gone to either essential or quasi-essential in that background. And so we added those genes back. Didn't work, but when we put wild type 5 in with the others, it was okay. So we then took that strain and made sequential deletions in segment 5. And we also added back 1 gene, which we found was required for vigorous growth. And having done that, we arrived at what we call JCVI SEN 2.0, 563 kb, 519 total genes, and that includes the RNA genes. And that's compared to the starting organism, which was about a megabase and 901 genes. This was the first time that we had something smaller than M. genitalium. And we considered publishing that, but Craig said, no, we got to do more than that. Anyway, so this shows the colonies. Here's the SEN 1 and the SEN 2. Growth rates are pretty respectable. SEN 1 grows about 60-minute doubling time, and this was 86 minutes. All right, so we did another TN5 mutagenesis, and we identified 37 additional genes that we thought we could now remove. And we also removed one of the ribosomal operons and a couple of vector sequences. And we then synthesized that, assembled the whole thing in yeast without testing the individual pieces, and it worked. We got a single genome. We called it SEN 3.0. And this slide kind of summarizes the overall steps. The yellow and the red are the genes that eventually ended up getting deleted. The yellow were the first stage, and then the red were the ones that we deleted going from SEN 2 to SEN 3. The blue were genes that we kept originally, and the green were ones that we had added back at an earlier stage. If you just compare the relative sizes, this is the SEN 3 cell. It's 531 kb and 477 genes, actually 473 genes. We didn't correct that number. And here, comparison of colony size. And the phenotype is a little bit ugly here. It tends to, instead of growing very nicely as separate cells of fairly uniform size, it grows in chains. The cells are variable in size, and it releases a lot of empty vesicles. In other words, it seems to be making excess membrane and pinching it off. So there's some defect. We are interested in finding out what that is. So the next thing was to look at the gene content. Chuck Merriman did a blast against 13 different organisms here. And he put the genes into 5 classes. The largest class here are what he called equivalogs. They're well-documented genes in the literature where there's no doubt about what the function is. The next group, 58 genes, were what were called probable. In other words, almost anybody would say we know what they do. Putative is still reasonably confident, but perhaps some question. And then we come to generic, where we have some information. For example, it's a transport or membrane protein. We don't know what it does. Or it's a hydrolase or an ATPase or an esterase or something else, but we don't know exactly what substrates it works on. Generic, 84, and then 65 genes, we don't have a clue. There's nothing in the literature, at least by blast analysis, hinting at what they do. We could set these into 30 different functional categories, and it's interesting. The ones in the bowl face are ones that are highly conserved. We didn't delete hardly any of these. They have to do with translation, replication, and so on and so forth, macromolecule metabolism. And then on the bottom here are ones that are largely dispensable. For example, there were 73 mobile elements in restriction systems. There were a lot of lipoproteins, of which almost all of them could be removed, and so on. To get a better picture, we partitioned the genes into 4 major functional groups: expression, of genome information. That's all the translational equipment, the ribosomes, everything, the tRNAs. And then there's a small number of genes, around 30 or so, involved in preservation of genome information. Those have to do with replication and cell division. Then you have a relatively large number of genes in the cell membrane. You have to remember that this organism does not have any real synthetic capacity. It can't make any small molecules. It can salvage amino acids, nucleotides, and so on, but it doesn't make them. And so you have to replace that by a very sophisticated transport system that can take up everything it needs from the medium. And there are about 120 proteins in the membrane, a substantial fraction of the total. And then there are some important systems in the cytoplasm, for example, energy metabolism, nucleotide, lipid, and amino acid salvage pathways, and so on. So that gives, and then there's this unassigned region where we don't know what they do. We assume that most of these, once we know what they do, will fit into these 4 categories. So that's about where we are. Where do we go from here? Our goal actually is now to have a completely defined cell where we know the function of every single gene. There are 84 generic genes of imprecise assignment, and there are 65 that are totally unknown. 149 genes. I think somebody enunciated the rule, one graduate student, one new protein function. We have to do better than that. So we're working on a strategy to identify those genes. We also need a defined growth medium. And we're very interested in having a systems computational model that completely describes the behavior of the cell. That's it. It took a lot of people. And work was supported by Synthetic Genomics, DARPA, and by the Institute. Thank you. Let me say one word. The paper just came out this afternoon in Science, and it's a free download. You can get the PDF. It's 17.6 megabytes.
Any questions?
So that paper should answer all the questions.